Now that we've had a look at the structures and the form of the renal system, let's have a look at the function and why these structures are so important. First up, we have filtration. At the kidneys, the blood undergoes the process of filtration. This means that some substances are passed through a semi-permeable membrane. This occurs at the glomerulus in the renal corpuscle, which, as you may remember, is the little bit that looks like this with the mixture of capillaries, and then the tubule goes on here. It is caused by a pressure gradient and a semi-permeable membrane. Let's have a look at that semi-permeable membrane first. This sieve, or glomerular filtration barrier, is made up of 3 layers. The first one is in the capillary and is called fenestrated endothelium. This is endothelium of the vessel that has windows in it. Fenestra is Latin for window, and allows some substances through but not others. Then we have a basement membrane, which we talked about in the lungs, which is a mixture of proteins and other substances but not cells. Then in the basement, in the Bowman's capsule rather, we have podocytes, where podo means foot. And these can be shown in this picture up here. You can see these large cells here with little outreaches that interconnect with other cells, creating tiny little slits that things can come through, that ions and water can come through, but protein cannot. And this is all over the capillaries. Each of these is a capillary under here, covered by podocytes. Here's another picture that shows the fenestrated endothelium with gaps, a basement membrane in blue, and then the podocytes, and you can see the gaps in between each of the outreachings of the cell. So these gaps allow through water, ions, glucose, amino acids, creatinine and urea. Creatinine and urea are waste products in the body. Waste. Does not allow through blood cells or proteins. And we'll discuss the importance of this further on. Now, I talked about the sieve, or the glomerular basement membrane, and podocytes and fenestrated endothelium. Now let's have a look at the pressure difference. The pressure that pushes the fluids from one side to another side of the sieve. First of all, let's have a look at this on the right, the hydrostatic pressure, which is the force a fluid exerts on the walls of its container. And in the blood is the same as blood pressure. So over here we have blood pressure. In the filtrate, we also have the force that it is exerting on the Bowman's capsule. So we have hydrostatic pressure over here. Then we have a thing called osmotic pressure. This requires a semi-permeable membrane, which we have with the glomerular basement membrane, a solvent, which we have water in the blood, and solutes, which we, in this case, are looking at the proteins in the blood. Now, osmotic pressure is related to osmosis. It is the force that the proteins apply to the water, a sucking force, to keep the concentration of water consistent across the semi-permeable membrane. So that is, the amount of water compared to what's in it is the same on both sides. When there are more substances in the water, we need more water molecules to keep the concentration the same. The water needs to dilute the proteins, is how I like to look at it. So what does this mean for the kidneys? It means that we have blood pressure pushing the blood this way, hydrostatic pressure in the filtrate pushing this way, and osmotic pressure in the blood pushing this way. Overall, we have two forces going this way and one force going this way, but blood pressure is the strongest of them all. It is the biggest force here. The blood pressure in the glomeruli is about 60 millimeters of mercury, so it's much higher than a usual capillary where it's about 5 millimeters of mercury. This force allows the filtrate to be formed, as it allows some particles and some water to cross the membrane. So we've talked about the blood pressure and how important it is in filtration. What's also important about blood pressure is that it is relatively constant in the kidneys. This is so that the flow through the glomerulus is the right speed. If it flows too fast, not enough will be filtered out, and if it flows too slowly, too much water and substances will be filtered out. If systemic blood pressure is too high, too much blood flows too fast into the glomerulus. So if it's too fast, we can't filter efficiently. Changes in the systemic blood pressure are counteracted by something called myogenic regulation. This is the vasoconstriction and dilation of the afferent arteriole, so the one that goes into the glomerulus. If our blood pressure is too high, the afferent arteriole constricts to limit the flow of blood. So we're getting too much blood coming in, so we get a narrower arteriole, which limits how much blood can come in at once, and this therefore lowers the pressure. If the systemic blood pressure is too low now, so we've got a low BP, the blood flows too slowly and too little into the glomerulus. If it's too slow, it will be over-filtered, and will remove too many substances and too much water from the blood. But then myogenic regulation comes into effect again, and the arteriole dilates. So we've got a little bit of blood coming in, but we have a dilated arteriole, so as much blood as possible is pouring into the kidney and the glomerulus. The glomerulus here. This will increase the local pressure because we will have more blood coming through, and more flow will increase more pressure.